Sticking with the theme of regeneration. Our bodies are designed with an incredible regenerative potential. Even now, we talked about a few tissues, but there are populations of cells called stem cells that you've probably heard a lot about. And so we're going to go over them because I want you to be familiar with this field and this space. Likely, you're going to be dealing with stem cell therapies as you treat patients in the future. So this probably will be an aspect of future medicine, if I had to bet and guess. Exactly what it's going to look like, I don't know. But it'll probably have some component of stem cell therapy that you're able to utilize as a healthcare provider. So kind of getting some fundamentals, and the field's going to change likely before you're out treating patients, but understanding kind of where it came from in the beginning. And since I've been teaching this class, it's changed. So, you know, it changes about every 5 years. There's something new of significance within young fields. But these stem cells that we have in our body, we have a larger population, a larger source when we're younger. We have, when we're in embryonic development, when we're in the embryo as a developing organism, we have the most. And for obvious reasons, because you're going to go to an entire organism from 2 individual cells. And then during childhood, we have a lot of hematopoietic tissue in our long bones, in the marrow space, and that gives rise to white blood cells and red blood cells. And that sort of fades off as you pass puberty because you're done growing and now you just need to maintain. So now our hematopoietic tissue at our phase in life is smaller in content. It doesn't occupy the entire cavity of the long bones anymore. It's only really up in the pelvic girdle at the ends of the femur, at that femoral head is where you find most of our hematopoietic tissue now, but it's still there. So we have circulating stem cells in our body, but they would be considered adult stem cells because we're all in this adult phase. So some images of, from marrow cavity space here and then circulating on the right, stem cells that are pseudocolored, but these are giving rise to replacement tissues in the body. We have a number of different types of stem cells. There's really 4 main types now. Okay, 4 main types, and I'm going to write them down up here. I'm trying to write them in the space where you guys can see. So the first main type that we have nowadays is embryonic. And the second main type that we have is IPS. The third main type is nuclear transfer. And then the fourth is adult. So we're going to talk about all 4. This slide only shows 3, and then the next slide will show the 4th one. So this is a great comparison slide. It's not that old. I pulled this out of the paper, and the field has actually changed a little bit since I got this. So I just added another slide. And that's just evidence to demonstrate to you that by the time you're done with your formal education, you're never going to be done with your education, but your formal education, this will probably change again. New things are happening. So in the embryonic category, this is our embryonic column right here. And this slide is kind of referring to this nuclear transfer as embryonic. I don't agree with that. I would consider this its own category. I would say that this is embryonic, this is nuclear transfer, and this is adult. But let's go through them real briefly. You can read sort of the attributes of each, you can read the limitations of each, and then you can read some of these ethical concerns. So in broad brushstrokes, real quickly, or briefly, our embryonic attributes can produce a cell, an embryonic stem cell. It has to come from an embryo. That's the reality of it. The most common source are leftover embryos from in vitro fertilization attempts. Okay, so a couple wants to have children, they're having trouble, they go to a clinic, they do in vitro fertilization. They take the egg and the sperm, and outside of the body, they allow for them to interact. Fertilization event is successful, and they cryopreserve those embryos. And then when the woman is ready, they implant usually multiple embryos because that increases the chance of success. If you implant one at a time and it doesn't take, then there's no pregnancy. So they implant usually a few. If all three take, then you have triplets, and that's what happens, okay? So then all three took. Mr. and Mrs. Smith now have triplets, but they paid, you know, $20,000 to this clinic to get 7 embryos, and they've only used 3. What do they do with the other 4? And they don't want their own TV program, so a lot of times they'll donate it to science, and that's where these embryos come from. Okay? So some of you are nodding your heads going, okay, that makes sense. Some of you are going, whoa, wait, time out. Okay, so here's where the ethical concerns come in. And these are tough conversations, right? Because everyone has different opinions. I'm just going to try to present to you what the facts are. But these embryos, if you use them for research, then they are destroyed. You can't use them for research and then go implant them and have a baby be developed. Okay, so usually it's after Mr. and Mrs. Smith have said, we're not moving on with any more, 3 is enough, not interested, but we don't want to just waste them. Okay? But you can imagine there's a population that is very concerned about that, and there's an ethical concern there. So that's sort of the negative against them, is these ethical debates or these questions. And maybe another negative would be it's actually rather expensive to put your hands on from a science standpoint. You can't just go to a catalog, you know, like Fisher Scientific, you know, order a couple of test tubes, some petri dishes, and like 5, you know, embryos, okay, for like $20. So a negative would be the cost and maybe the ethical concerns. The pros. It can produce essentially anything. You can produce all cell types. You get a whole organism out of the embryo, one cell. So if you have an embryonic stem cell, you can make anything. So it has what we call totipotency. It has an infinite potential to become any type of cell or any type of tissue. And there's a lot of power there. They're relatively easy to grow in the lab. These cells kind of want to do their thing. They're programmed to grow. That's what they do in utero. Some of the limitations, some more of the cons, that second row. There's a limited pool of available blastocysts or stem cells, embryonic stem cells. Federal funding to support embryonic stem cells is sort of lost favor. There's a huge risk of forming teratomas, which is another name for a benign cancer. Many times in the literature, when you see scientists utilizing embryonic stem cells, it's almost like there's too much power that we don't know how to control yet as humanity, and you form sort of a mass of cells that have like hair and bone and teeth and nails, and it doesn't work. Okay, so these teratomas, they're considered tumors. They're typically benign, but if you implant them into a patient to try to regenerate cardiac tissue, there's no guarantee it's going to become cardiac tissue. Make sense? All right, far right. The polar opposite are adult stem cells. And we're going to talk about 2 versions in between. So embryonic and then adult. Adult derived. Let's talk about our ethical concerns. We haven't seen any ethical concerns raised about adult derived stem cells. You don't. It's not destructive to the organism. We've been doing bone marrow transplants for decades in patients. Arguably, if you donate blood, you've donated your adult-derived stem cells to somebody, and it didn't kill you. So there are really no ethical concerns that I can think of, and that the field has ever said. Okay. Limitations, though, on this column, working our way up, they only can produce a limited number of cell types. You can't create anything. Because they're not embryonic, they don't have all the power of the embryo, so you're limited to certain types of tissues and cells that you can actually generate. You don't see them everywhere. You don't find stem cells in the adult in every organ or every tissue. They're only in certain places in the body. We don't see them in neural tissue at all. That's why strokes are so problematic. They're a little difficult to identify and isolate because you don't get a pure population. And so they can be a little finicky to grow in a lab. Take a little bit more energy. Their attributes, the positives. We've used them as a field successfully in numerous treatments. So there is actually a history of using stem cells, adult-derived, like bone marrow transplants. Okay, we've been doing them for decades, for 30+ years. The stem cells can be genetically matched to the patient. That's what happens with a marrow transplant. Now, the other thing is now we know that we can take cells out of ourselves, cryopreserve them and store them, and go back and use them in the future if we need it. So we could actually be our own donor for years later. That would be autologous stem cell donation to yourself later in life. Okay, so now would be the time to do it when you're younger. You know, if you're 60 years old and you donate and you cryopreserve your cells, and then something major happens at 70, those cells are only 10 years different. Does that make sense? That's why you have all of these companies that are trying to cryopreserve stem cells from the umbilical cord or from the placenta. Because those are not embryonic, actually, but they're extremely young adult-derived stem cells. Right? They have a lot of attributes like embryonic cells because they're so young, but they're still technically not from the embryo. Okay? So that's that whole industry of cryopreservation of stem cells. Yeah, what about this middle one, Keller? Well, there's two middle ones, and these ones are really kind of clever. These two are clever because they're really not either adult and they're not embryonic. This one's called nuclear transfer. There was a Korean laboratory that sort of popularized this one. And then they got under fire because it said that they falsified their data. Okay, so they were the ones that came on the scene initially, and then they just like crashed and burned. Okay, and then others came after them and did the nuclear transfer. So what this is with the nuclear transfer is you actually take the nucleus, right, and you put it into the donor cell, and it becomes a stem cell. So you take a nucleus out of the recipient, and you implant it into a donor cell, like a oocyte. Makes sense? So you need donor oocytes, which isn't destructive to the organism. It's actually rather painful. It's a lot more complicated for women to donate oocytes than for men to donate sperm. Okay? So the oocyte donated by the female can be transfected with the nucleus of the recipient that you're going to actually give it to. So now you have a nuclear transfer stem cell that's embryonic-like. Are you with me? Because it came from an oocyte. So let's look at the attributes. You can produce all types of cells, relatively easy to grow, maintain. They can be genetically matched to the patient, just like I explained. Some of the limitations, again, there is a risk of creating teratomas. It's not quite as big as this one, but it's still there. And the ethical concern is the destruction of an oocyte, really, is what it is. So it's not quite to the same level of concern as this one. The donation of the egg requires informed consent, right? You don't just randomly walk up to a female and say, I'm grabbing some eggs. Okay? And there's some concerns about cloning. This is where the cloning concerns showed up on the scene. Human cloning, okay, with nuclear transfer. This was the, do you remember the Dolly experiments? The sheep? Okay. So that's actually this process of nuclear transfer. This is where the cloning, human clones, would come into play. Okay, so what's this IPS? This IPS is a little different. So this IPS one is on this slide that I took out of a different paper. So here's our embryonic stem cells, and this kind of shows the process we talked about. Here's our adult stem cells, and you can see in the category of adult, umbilical cord, placenta, amniotic fluid, bone marrow. I'll show you fat. We can get stem cells from fat. So these are all considered adult-derived stem cells. This one that we haven't talked about is our, I guess it's our second one on the list. This is our induced pluripotent. So you take a normal cell and you transfect it with certain genes, or you utilize certain chemicals that essentially cause a transformation of the cell to behave more like an embryonic stem cell. And you induce what we call pluripotency. So they're called induced pluripotent stem cells, IPS. Pluripotent means that the cell has the ability to become many different types of cells or tissues, but it's different than totipotent, meaning totipotent is that one cell could become anything. So embryonic stem cells truly are totipotent, and the IPS cells are more pluripotent in nature. Okay. Adult stem cells, we would consider them to be multipotent. Not pluripotent, but multipotent. They can become a number of different things, but as you move further away from the embryo, you get less variety that you can generate out of the stem cell. Make sense? So this truly is going to be in medicine. It's in medicine today. There are clinical trials now where patients are getting stem cell therapies, not just bone marrow transplants, which have been going on for years, but they're getting stem cell therapies like injected into their infarcted heart muscle. Okay? They're getting stem cells post-stroke intravenously fed. Most of these types of therapies are happening outside the United States. Okay, it's very popular for these clinics to exist in South America or over in Europe. The clinics are attached to hospitals and kind of like a resort where you can recover. It's usually for the rich and famous or the very, very wealthy. And they'll go there and they will have their therapy and they will come back recovered in miraculous ways, but you can't do it within the borders of the United States quite yet. Okay, a couple questions.
Can you say the definition of totipotent?
Totipotent is the cell can become anything, the whole total organism. Pluripotent, pluri meaning many, the cell can become many different types of cells or tissues. And then multipotent is it can become a limited number of things, but not just one. Okay? That help?
Is it the same teratomal risk for IPS?
As what?
As embryonic?
No. IPS has a risk of teratoma formation, but it's less than what you have with embryonic stem cells.
What makes the embryonic stem cells way more specific to be able to go to any type of cell, like IPS and certain types?
Well, the question is, why is the embryonic stem cell able to become so many things? Why is the IPS cell essentially limited? It's a great question. I don't know. I mean, the embryo is so much. I mean, if you take a moment to pause about what happens in 9 months, you know, for a life to be made, it's unbelievable. And so the amount of energy and power and creativity in that, from a cellular standpoint, is huge. And that's pre-programmed. You know, moms work hard, but it's not like every day they're like, all right, let's see, vascular system, bam, right? All right, let's get some legs out today, yeah. You know, I mean, they don't have. I gotta be careful here. They're doing a lot, but they're not thinking about the process. Does that make sense? The process is happening, so the cells know what they're doing. So that programming is there. That programming is in the DNA of the embryonic stem cell. And in the IPS or in nuclear transfer, we're fabricating that. Man is fabricating that.
So we don't really know why they behave the way that they do.
Yeah, that's correct. We don't really, that's how I started my answer, right? I don't know. We don't know why. We just observe what happens, but we don't necessarily know why. But these stem cell therapies are now making their way into MS patients, multiple sclerosis patients, stroke patients, cardiovascular infarct patients. A lot of this is happening outside the borders of the U.S. and in some limited cases in clinical trials in the United States. but by the time you all are treating patients in the clinic, I imagine it'll be a lot more prevalent. A lot more prevalent.
So is that mainly because of like FDA regulations?
Oh, it's absolutely because of FDA, yeah.
So it's mostly political, not necessarily because of risks?
Well, I mean, FDA isn't completely political. I mean, so they're, you know, it's a yin and a yang kind of a thing. I mean, there was a season in our country before 1924 where the FDA didn't exist. And the real short story, and then we got to move on. So the FDA didn't regulate it like they do today. And there was a antibiotic medication that was very, very effective in treating infections. And this is in the southern part of the United States. And the company that was making and selling that product wanted to make a pediatric version so kids would, it's very bitter. And they wanted to make an oral solution that would be taken, and so they put it in a glycerol-based solution, and it was very sweet. But the formulation that they made is the same formulation that's in antifreeze. And so they had children dying, and that's when the government, the president, said, TV timeout, and we're going to put some real teeth into this agency, and that's when the Food and Drug Administration was born. So it was a tragedy that it's usually the case, right? It was a tragedy that caused this event, and so you know, it's this pendulum. Regulations are good, and then they over-regulate, and now you stifle progress, and then you know that comes back to the way less regulation, and then people get too creative and too relaxed, and something bad happens. So it's kind of somewhere in between is probably good. But the reason, to answer your question long-windedly, the short version is the Food and Drug Administration has a process to get things through, and these things have to go through the process of clinical trials. Okay, last question, and then, okay, all right. All right, so I want to talk to you about some work that actually we do in my lab. It's kind of fun work, and it's very relevant to this stem cell concept. We use adult-derived stem cells. We don't have any ethical concerns, and in the Western world, there's plenty of fat around, okay? And most people are willing to give it up. So we take adipose-derived tissue from liposuction from the patient. We do this aspiration and this wash with the digestion with collagenase to get rid of the collagen. Do a few different types of spin downs and essentially isolate what we call a stromal vascular fraction. That's the pellet. We grow that out in culture. That contains the stem cells, not only stem cells, but it contains mostly stem cells. And those adipose-derived stem cells, those ADSCs or the ASCs, they can differentiate into 4 main lineages. That's why we call them multipotent. Because they can become fat, obviously, that's where they came from. They can become bone, they can become muscle, and they can become cartilage. Okay? Now we're seeing there's a 5th line where they can actually become fibroblasts. And that's not on this particular slide. This is an older slide from 2009. So now we see a 5th line of fibroblasts. And you can put those into numerous clinical applications, and that's exactly what we are trying to do. So the first thing that we do is we build a matrix. Right? So we build a matrix because I just told you that in all tissues, it's a matrix in cells. So this is a few students that built this. This is an electrospinner. Proteins are chargeable. They're usually negatively charged, but that means that you can charge them positively or negatively, and they will carry a current. So this is our high voltage power supply. This is our target, which is our negative ground. This is our syringe pump with our needle. In here is a solution of protein into a tubing onto a needle, and this is a positive electrode. And then this creates, it's like making spider webs. It's like spinning spider webs. I'm going to show you a video real quick that characterizes what this looks like. This the setup in the lab. It never looks this pretty or clean. This was photo day, so there's no stuff laying around. But here's the syringe pump right here. Here's our power supply. That's a 25,000 voltage power supply. This is the acrylic box. Here's the needle, and then here is our copper mesh, which is our target. And it's kind of like spinning spider web, so it looks something like this when it comes out. And that's going down here on this platform, and that's going to try to replicate the matrix environment of tissue. In our case, in this application, it's actually skin. So this is what we get. We get this matrix. It looks like a jungle gym, right? So it's got all these architectural fibers, and on the left side, this is a fast flow rate, and on the right side, this is a slow flow rate. So if we inject the solution slow or fast, we can change the density of the matrix, and we could potentially match it to skin or liver or heart or the environment that that we're trying to replace. In our case, it's skin. Well, we take that matrix and we grow stem cells on it. We take fat cells from humans, and we get these stem cells. Here is a mitotically dividing adipose-derived stem cell. Down here, you can see the bare elastin scaffold. This protein we spun was elastin. That's the bare scaffold down here. This is the dividing stem cell. You see this little bump? They spit out matrix when they are happy, when they're living in this environment, and it's kind of like little kids in your bed with the sheet pulled over them. You know what I'm talking about? So Saturday morning you come in and it's like, oh, where are the kids, right? And you hear this little wiggle, giggle, right? And then you jump on top of them. And so that's like these little that's when they get their injuries, the wounds. Study, so it all works out. Lamp hits them in the head, causes a lesion. So this is the little bump right here of the cell, and all around it is the matrix. So this is our patch. This is a wound healing patch. We made a matrix, we tried to do skin, we impregnate it with stem cells. Now these are human stem cells, and so we wanted to implant it into an animal model. That's to the FDA question. The FDA will not let you move into humans until you've treated animals and made sure that it's safe and it works. Okay, so this is the first phase of that. So we made these electrospun scaffolds using these proteins. We got stem cells from humans, and we want to put them into a mouse. Well, you'll see in a couple of weeks that's a big problem, because the human will reject the mouse cell, and the mouse will reject the human cell. So we used an immune-compromised mouse. And we'll learn about that a little bit more in a couple of weeks. These are severe combined immunodeficient mice, so they don't have an immune system, so they don't see a foreign cell. They don't even know that it's not theirs. We went to 6 days. We had a control group. The control group wasn't just an open gaping wound that we spit in and stomped on just to see that it was yucky. We actually used clinical standard of care. So this is, if you went into the hospital today, you would actually get Vaseline gauze and a dressing known as Tegaderm covering the wound. So that's what the control group had. So here's an example of what the wounds look like. We made these full thickness wounds all the way through the skin. This is a grass. His name's Hans. He's actually in med school now, but he was doing these surgeries. He was growing these scaffolds. He's picking them out and placing them onto the wounds, and this is after 6 days of healing. So here's our control. If you can appreciate the green dotted line, kind of is tracing the open wound still. So after 6 days of healing time, these wounds were, maybe 60 to 75% healed. These wounds were almost 100% healed. This is the scaffold plus the stem cells. This is just a different type of scaffold. This is elastin, and this is collagen. We tested 2 different scaffolds, and then the same stem cells. And these were basically the same. They were pretty much all healed after 6 days. So if you look at graphically what this looks like, this is a representation of the wound closure. You can see that the control therapy works pretty good. After 6 days, 75% wound closure, but we were in the low to mid 90s as a percent closure only after 6 days of therapy with these stem cells. So the capability of stem cells for wound applications is actually rather huge. Another metric we measured was epithelial thickness. The thicker the epithelium after 6 days, then the more aggressive the healing response was. So the two scaffolds with stem cells were statistically thicker than the control group as far as microns of epithelial thickness. That's only after 6 days. So that would be 2 weeks later, you would probably see something very different, which is something that we still have to do. So I'm using this as an example to talk a little bit about how wound healing takes place, how it works, why I think we're going to see applications in the near future. And it's not just going to be with skin. It's going to be with all different types of tissues. So in the last little segment, I want to talk about some more clinical applications, and I want to tie in these different phases of wound healing. We've got these three main phases we've talked about: inflammation, maturation, and inflammation, proliferation, and maturation. Well, they also have some other terminologies that are associated with it: the coagulation phase, or the migration proliferation phase, or the remodeling or the maturation phase. So you might see these 6 different terms used to describe these 3 phases sequentially of wound healing. In our coagulation or our inflammation phase, you've got breaching of the epidermis, and you have blood vessel damage. That's going to lead to a response, right? That response is going to be a fibrin clot, right? You're going to get vasculature responding, cells being recruited to that area. This is a little bit of a review of where we've been. Well, the next phase is this migration or this proliferation phase. Now we start seeing the specific cell types moving or migrating to where they need to be. You see proliferation of stem cells or fibroblasts in the area to repair the cellular damage and to be able to crank out new collagen to repair the matrix damage. Migration and proliferation phase. The last one's our remodeling phase, and you can see the scar that forms. Well, we saw this in the first and second intention pictures. But this is where the tissue remodels, because now it's remodeling. That kitchen remodel, the kitchen doesn't look like it used to. This wound remodeling, that remodeled tissue doesn't look like it used to. Right? And this is where it matures, and it's going to essentially stay in that consistency forever. So let's look at some other clinical examples. I'm going to put a little warning. You might see the picture there. Sometimes it's a little bit more graphic on the big screen. So just for effect, and to ensure that maybe somebody vomits, I'm going to turn off the light. So this is a full thickness dermal wound, obviously across the left cheek of this patient. And the best treatment, approximate those wound borders with sutures as soon as possible. Okay? I don't have a feeling this wasn't Saturday morning, bugs in a bed exercise. This was something a lot more aggressive. Okay? Well, sometimes we can't approximate these wounds. Right? What do you do here? Wound approximation is not an option. This is a burn victim. So when wound approximation is not an option, we have some choices. We can do autologous skin grafting. We can use cadaveric tissue, which would be an allograft. Allo meaning same species, right? Or xenografts. That means different species. Like you can get it from pig or bovine, which is cow. And you take the cells out. You strip the cells out. You just treat it with the matrix as a patch. Okay? So these are our options that we have. There are some commercial dermal substitutes. For example, this company started it. I went to work for these guys back in the late 90s. And they're not around anymore, but they sold off all their, we ended up selling off all their technology and piecing it out. And the technology exists today still. The company that actually sells the wound product I'm going to talk about, Dermagraft, is actually in Boston, and their company's name is Organogenesis. So this Dermagraft technology is where I kind of came from. So you can see that our experiments we're doing now are a little bit sexier because we're using stem cells. I think stem cells are sexy, but you know, that's how nerds are. So, but maybe a little bit more interesting and new, but still the same concept. We take a scaffold back then. This was a bioabsorbable scaffold. We seed it with fibroblasts, dermal fibroblasts. We got the dermal fibroblasts, the skin fibroblasts, from very young throwaway skin. Any ideas where that came from? Circumcised foreskin. Right? Just goes in the red bag. You don't need it. You don't put it in a scrapbook usually. Okay? I mean, usually. Those donors we didn't get because they, you know, if there was a scrapbooking family, then, you know, I guess they save everything. So we would get the leftover throwaway circumcised foreskin, and we would harvest fibroblasts out of it. How's that for committed to science? Huh? And we would grow them on this scaffold, and they would make their matrix and their growth factors. And they would create this replaceable tissue. Doesn't that look eerily similar to what I showed you just previously? But this was back in the early to late 90s this is happening. Okay? Well, here's how it was used for treatment. This is a diabetic foot ulcer that I was telling you about. This was a clinical trial. I think the date's on there. Where's the date? 95. Do you see the date there? 12/12/95. Right? So you can see the date. This is 10 of 95, or 10/19, 10/24, 11/7 of 95, 11/21. So 10 sequential weeks. Okay? These patients, to get into this clinical trial, had to have a non-healing wound that was open for 6 months. So it looked, this is the bottom, you're like, what am I looking at, by the way? This is the heel, that's the bottom of the heel. Yeah, would that suck or what? So 6 months they dealt with this sucking wound, which is a very clinical term. And they got enrolled in this study. After 1 week of this patch being treated, after 3 weeks, after 5, look at after 8 weeks, you have complete wound closure. Right? So we were pretty wound up about this. This is cell therapy, not stem cell therapy. This was like the precursor to stem cell therapy. And these technologies are being sold today. Dermagraft is on the market today. It's being used to treat diabetic foot ulcers today. We also discovered that it was very angiogenic. Well, what does that mean? Well, if you look at the different weeks, this is 1 week, 3, no, I'm sorry, this was 3, 5, and 8. 3, 5, and 8 weeks. If you look at the tracing, the blue are the cool colors, that means no blood flow. The tracing of the wound here, now you're starting to see some brighter colors, blood flow. Look at after 8 weeks, you've got lots of blood flow into the wound itself. So it was angiogenic, right? We did some experiments on angiogenesis. We looked at the production of vascular endothelial growth factor, remember VEGF? In a 2-day period, we started seeing ramp-ups of VEGF production. Look at these little experiments over here. This is the control mesh, no cells. This is the mesh with the fibroblast. These are all new blood vessels that grow into that mesh as a result of those cells being around. Question?
Is there a point at which the dermagraft doesn't work?
Is there a point at which, I mean, there are some wounds. I'm going to show you an aggressive one here. There's probably been over 60,000 patients treated with dermagraft as of today. You know, I don't do anything with the technology anymore. We sold it and moved on, but I'm sure it has limits. I just don't know what they are, to be honest. Okay, I'm going to show you a pretty crazy example here to finish us off today. So this is a warning again. If you're going to hurl that way. Okay. This was a accident with a chainsaw. Yeah. Accident with a chainsaw. This was up at UCLA, and that's a full thickness wound. That would classify as a full thickness wound, no question about that. Very few people would argue with that. This was after closure. Approximate the wound. That's a pretty good closure right there, by the way. Right? Can you notice that? Okay, so that was a traumatic wound. This is healing by second intention or first intention? What do you think? With a chainsaw. If there was a third intention, I'd pick that one. We don't have that option. So this is second intention. Look at how nicely the wound has been approximated. You with me? And then dermagraft was attached, okay, and used and treated. This is the patient a year later. Handsome dude, huh? So take a peek. Don't shout it out. Take a peek. I want you to tell me, looking at this picture, is this wound resolution or is this wound repair? Is this wound regeneration or is this wound repair? Don't say it out loud. Talk. Bet with your neighbor. All right. What do you guys think? What's the voice of the crowd? It's resolution and regeneration, isn't it? Why? He's got function. How do you know he has function of that organ now? Because the hair, that's why we're like, dude, grow a mustache, man. You know, just rock a mustache and show everybody you got total regeneration. Okay? Pretty cool, huh? All right. Any questions? Any last remaining questions?